Good morning, everyone, and it's a pleasure to welcome Professor Athi Naganathan from IIT Madras. Professor Naganathan is now Associate Professor in the School of Biotechnology, IIT Madras. Professor Naganathan obtained his completed his BTech degree from Anna University in Chennai before going to University of Maryland, USA for his PhD. So he worked with a very influential protein theorist slash experimentalist, Victor Munoz, and followed by his PhD in University of Maryland, he moved to Spain. Was it Madrid first and Madrid first, and then to Barcelona, where he continued working on the field of protein folding. So, and he has set up this his group, his laboratory of protein biophysics in IIT Madras in 2012, and he has quite a few awards, including the INSA Medal for Young Scientist and one of the prestigious fellowships in India, the Wellcome Drugs DVD India Alliance Fellowship 2015. It's a pleasure to have you here. The floor is yours.
Thank you, Kalyan, for inviting me here. And the first time for me, I'm here, and very nice place. We're having interesting discussions along the way. And I'll tell which one. I'm not going to disclose that for now. So, I'm Adi, and this is my group website. If you're interested, you can look into it. I am from IIT Madras, and the title of my talk, as I have written here, is something on homo versus hetero-oligomerization of proteins, protein DNA complexes, and how they could kind of drive thermo, meaning temperature dependent, and osmo, which is effectively changes in ion exchange conditions or salt concentrations with bacteria base when they infect, say, a human host, and how they could, these kind of proteins that we are going to talk about, can drive these responses. And we are not going to, or at least here in this work, I'm not going to talk anything about in vivo studies, all in vitro purified proteins. And the idea of this work to see what happens when you take some of these very interesting sensory proteins in bacterial systems and you reconstitute them in vitro and you do basic analysis of these. What do you obtain, and what does it tell you about the functioning of proteins that comes with it, right? So to start off with, just to kind of briefly introduce what we do in our lab. We do a bunch of experiments, right, which can be CD fluorescence, fluorescence lifetime, FRET, ESC kinetics, and so on and so forth. We also do all atom and post-grain simulations. We also do simple basic statistical models. It's very, very simple models that we deal with. And combining these, which is combining these, we kind of generate some folding conformational landscapes of proteins. Kind of comfortable with some terminologies here. These landscapes carry information on the barriers and reaction coordinates, intermediates, and the functional constraints that proteins have kind of evolved into, and the time scale of some of these conformational transitions can also be obtained. And you will get a feel of the heterogeneity, folding, binding, and function. That is something that's coming up more and more, and on how some of these very interesting systems, particularly DNA binding proteins, can form higher order macromolecular assemblies. And of course, now, which is a big thing in biology, which is molecular condensates, which is liquid and liquid phase separated droplets. Some of these can form, and hence you can kind of build in some nanomaterial like architectures in these proteins. And then, of course, we are also interested in engineering proteins and in predicting structures, aside from I had before, but now no longer interested in predicting structures over the last 2 years or so after. Right. So, just to go to the theme of my talk, and is how do bacteria sense environmental? For example, we have a bacteria in the soil, and it need not express many of the proteins that it generally expresses in itself. On infection, it needs to express other proteins. In the soil, or if it's not infecting a human host, for example, it need not express other proteins. So, but it needs to sense if it has infected a human host. So, how does it go about doing it? Right, and there can be many cues, and something that I'm more comfortable with is some thermodynamic variable like temperature and something like a pH. It's called obvious, and of course even the osmolarity, which is the salt concentration within the human. Right, there can be very different sensing mechanisms. There's something called non-protein based sensing, meaning protein molecules are not involved, and in here proteins are involved. And when it comes to non-protein based sensing, changes in temperature, pH, or osmolarity can change the DNA topologies into being a circular DNA to kind of a supercoiled DNA, or you can have conformational transitions in RNA. For example, in this case, this RNA might not be translated, but when the temperature is on the higher side, what happens is that these interactions are lost and the RNA kind of unfolds, and hence this RNA can be translated. This is a known mechanism when it comes to RNA. And this is also it's very obvious when you have membranes. Membrane fluidity can be altered, and the changes in membrane fluidity, particularly due to temperature, can be sensed by downstream response elements, and hence expression of certain genes can be initiated. Right, and what we are interested is on the theme of protein-based things, that when you have a protein molecule that is kind of floating around in the bacterial cell, and the bacteria is about 25 degrees Celsius, now you suddenly increase the temperature to about 37 degrees, which is what happens when it infects a human host. Maybe the protein has been designed or is evolved in a manner that it's able to kind of unfold. Whatever reaction or catalysis that it was able to do at that particular temperature, low temperature, it would no longer be able to do at the higher temperature. That gives you a very simple way to actually control function. Right? The other way to control function is that at low temperature, protein could be oligomeric, and each of these circles could be a monomer, and eventually at higher temperatures, these oligomers could disassemble, and hence you can control function. For example, you know this because many of the transcription machineries is collection of oligomeric assemblies, protein DNA complexes is what you see there, right? And this can be homo oligomers or this can be hetero-oligomers. Two different types of proteins can come together at some low temperature, and as you go to higher temperature, some of these can kind of disassemble. And this is what we will be talking about more so today in this lecture, right? So here, these changes, conformational changes in DNA, RNA, membrane fluidity, or even proteins at the level of either a monomeric protein or in terms of assemblies can turn on or off a certain gene, and hence the bacteria is kind of able to express certain genes. It helps to survive in that particular condition, right? So that's the way bacteria generally go about doing things. And what we are interested primarily is how do proteins act as sensors of ambient conditions? Can we learn something from a natural sensor, which is a protein called Cnu? And how do protein-based stress adaptive mechanisms really determine bacterial survival adaptation here, right? And this can help in design of higher-order molecular complexes and potentially drug discovery. I'll come to this at the later part of this talk. This is something that is published, and this is something that we're currently working on with some interesting observations, right? So the simplest sensor in a bacterial system is this particular protein called Cnu and H-NS. This Cnu is a monomeric protein, which is about 70 odd residues in length, and H-NS is about 110 residues odd in length in terms of the number of residues, right? So this is considered as a co-repressor, and this is the repressor in the sense that H-NS is the one that binds DNA, and Cnu controls the extent to which H-NS binds DNA, and hence it's called a co-repressor. What has been established through biochemical experiments is that at low temperature or low ionic strength conditions, in other words, low osmolarity, what happens is that somehow Cnu and H-NS are able to interact with each other, form some functional complex. They're able to bind DNA and repress the expression of genes. Right, and hence, say for example, if the bacteria is not infecting a human host, this is what is likely going to happen, and hence you don't want any unwanted gene expression, right? So, but when it infects a human host, say, and the constant body temperature of 37 degrees Celsius, 3 times Kelvin, and at higher ionic strength conditions, which is the kind of conditions that you see in the human network, what happens is that for some reason these two guys are not able to see each other or interact with them. With each other, and hence the bacteria is able to kind of escape from this suppressed state and start expressing genes, right? So this is the basic system that I'm going to talk about here. Nothing within the cell, as I said, only in vitro and purified proteins that we are working, right? So what we did was, this is the protein called Cnu, this protein which is called Cnu, and we wanted to see what does this protein look like. Is it actually sensing changes in temperature? Is it sensing changes in pH, osmolarity, and so on and so forth? We wanted to understand what happens to the co-repressor before going into the repressor, because repressor purification was a big problem for us. We weren't able to purify H-NS, but we were able to purify Cnu reasonably well. And what we find is when you look at the structure, there are many aromatic probes here. And when you do a simple melt of this particular protein as a function of temperature, so what you have is temperature on the x-axis, and this is some measure of the tertiary structure out here. And what you can see is, depending upon the wavelength, We monitor at 280 nanometers, you can see the signal increasing, reaching a maximum and then decreasing. But you look at a different wavelength, what happens is the protein shows a sigmoidal-like unfolding transition. Generally, when you take any protein, you will see this kind of sigmoidal unfolding transition. Here the protein is fully folded, here the protein is unfolded, and anything in between is probably a combination of folded and unfolded molecule.
What is this wavelength of optical?
It is a near UV circular dichroism. It's a circular dichroism. Right. So at 280 nanometers, you primarily monitor tyrosines and tryptophans together, and 290 nanometers, you primarily monitor. This is what happens. Right, and you monitor this particular tryptophan at 290 nanometers. At 280 nanometers, you'll be looking at a combination of all those. This is what you will be able to monitor. And when we looked at this, we weren't able to understand what the heck going on with the system, because we didn't know. Generally, you would see only a sigmoidal transition when it comes to proteins, but here what you see is not just a sigmoidal transition, but also another scenario where this thing increases and decreases. Generally, when you are in the protein folding field, this would effectively signal the population of an intermediate. Population increases and then drops down and the protein molecule starts to grow. So that's what this means. Right, and when we monitor only the tryptophan, this particular tryptophan here, there is only one tryptophan in this particular protein. So as a function of temperature, when we monitor the quantum yield of this particular tryptophan by fluorescence measurement, by exciting it to 95 nanometers, we get this nice transition here. And the midpoint or the inflection point of this particular transition, which is somewhere here, is about 310 Kelvin, which is significantly lower than the inflection point of these transitions, which are about 330 Kelvin. So whenever the signal changes earlier, it suggests that that particular structural element is actually melting early, since it unfolds early as a function of temperature. And since this is somewhere in the physiological range between 280 to 290 and 310 Kelvin, is the physiological range. What we think is happening is that maybe this particular tryptophan is actually unfolding at the lowest temperatures, and hence you have a strong change in signal even at the lowest temperatures. Right. So this is our original interpretation. Right. So and this is something that we weren't sure of. You have an observation, we need to explain this observation, and so we resorted to the so-called Walker-Saito-Munoz-Eaton model. It's a very simple model. It's Go-like in the sense that you take a protein molecule, what you can do is that it will assume that those interactions that are present in the protein molecule are the ones that are driving the folding of this particular protein or the folding mechanism. Right? And it's coarse-grained at the residue level in the sense that we have information at the level of individual residues or not at the level of atoms, unlike molecular dynamic simulation where you have information at the level of atoms and you accumulate conformations as a function of time. You do it for 100 microseconds or 100 nanoseconds or several tens of microseconds. Here we don't do this as a function of time. It's purely a thermodynamic model, which we assume that each residue in the protein can start to two different conformational substates. It's very simple to just say one for folded and 0 for unfolded. So which means for
when you say it's unfolded,
yes.
Like the telephone coil is
Unfolded, yes.
Unfolded.
Right. Yes.
Mathematically there are some invariants associated with that. Are you looking for them or only observationally you see that it's unfolded?
No, so these are models where we I'll come to that in a minute. So what we do is we use this model to explain or try to understand some experimental data that we have. That's all we use this model for, not for anything that develop invariants. But for example, I'll just go through this maybe at the end of this series of slides. Maybe we can have a more detailed discussion. Right? For example, if you have a 3-residue peptide, we are interested in asking, okay, how many different conformations are possible for this 3-residue peptide? Right? And assuming that there are 2 conformational substrates for every residue, and I can label this is residue 1, this is residue 2, every column corresponds to one residue. This is residue 1, this is residue 2, and this is residue 3. And these are the different conformational substrates that are potentially possible, assuming that it can be 0 or 1. A fully unfolded peptide would appear as 0, 0, 0. A fully folded peptide would appear as 1, 1. Right? And any combination of 0 or 1 can represent potentially partially structured states. If we know the statistical weight or probability of each of these different states, then you can actually go about modeling this particular unfolding transition that I showed you in the experiment. Right? And that's where this model comes. This model was actually developed by Walker and Saito back in 1978, and it was actually lost to time. And Munoz and Eaton, they kind of independently came up with this model back in 1999, 1998. And then Munoz, with my PhD advisor, was this model to me. With We have been working on more complicated versions of this particular model over the last 10 years or so, right? So now, of course, we cannot say that 0 can represent one state of a molecule or a residue, because an unfolded like conformation can sample many microstates, can sample many states, while folded means it's fixed or rigid, the particular environment of the protein. So we kind of introduce something called entropy penalty, which is effectively telling you what kind of penalty that you need to provide to the system to ensure that the molecule goes from the unfolded state to the position. You need to provide certain energy to the system to transition from 0 to 1. And that's what we kind of introduced via this entropy penalty. And of course, if you can imagine, if it's an end residue protein, you're going by this argument, you will have 2 power n microstates or conformational substates, if I can say, n being the length of the protein, right? So, and the primary assumption of this model is that there are 2 islands of ones. For example, there is one island of ones here, there's another one here. These 2 can interact only when the intervening residue is also 1. In other words, this particular conformational substrate, these 2 ones don't see each other. For the kind of transfer matrix formalism that we use, because we cannot do or look at the statistical weight or calculate the statistical weight of, say, 2 power n microstates. If you have a 100 residue protein, you will have 2 power 100 microstates, which is a very large number. You cannot algorithmically enumerate it. You need to use something called the transfer matrix formalism, which I'm not going to get into here because that's not the point of the talk. But you can, to use the transfer matrix formalism, you would need to make some assumption as to how these kind of residues interact. And the assumption here is that if there are 2 islands of 1 separated by zeros, these 2 islands can interact only if the intervening residue is also a 1. In other words, you need to have a continuous stretch for the interactions to happen, right? So, how do we use this particular model? And as I said, there's 3 residues: residue 1, residue 2, residue 3. We can label each of these conformational substrates as A, B, C, D, E, F, G, and H. And if I have the statistical weight of each of the states, statistical weight is some kind of measure of probability, right? So now I can calculate what is the probability of my first residue to be folded. What I can do is that I can take the statistical weights of those states in which the first residue is folded. You can see state D, the first residue is folded. F, first residue is folded. G, the first residue is folded. State H, first residue is folded. I sum of the statistical weights of those states alone, normalized by the total partition function, which is the sum of the statistical weights of all these different states, and I get the probability of the first residue. And similarly, I can do it for residue 2 and for residue 3 because there are only 3 residues. Right? Similarly, I can do or also generate something called free energy profiles here. And the way it works is that instead of looking at which residue is folded, I ask the question of how many residues are. This is some measure of reaction coordinate, or in terms of some kind of order parameter that we look into this particular system. And here you can see that there are no residues are folded. N is equal to 0 here, N is equal to 1, N is equal to 1, N is equal to 1, N is equal to 2. We need 2 residues are folded, 2 residues are folded here, 2 is folded here, 3 folded here. Now I ask this question: what is the probability of identifying no residues to be folded? What I do is to take the statistical weight of this state and divide by the total partition function that gives me this P. And similarly, I can do it for what is the statistical or rather, what is the probability of identifying all the 3 residues to be folded? I take the statistical weight of only this state and I get the probability here. And similarly, I can get for 0, 1, 2, and 3. I can do it, it's 100 residue protein, I can do it for 100 so-called microstates, rather macrostates here. And now I can convert this probability into free energy value minus RT log P, and I can just project them onto the one-dimensional coordinate, and that will look something like this, which will tell you what happens as a function of the reaction coordinate, how much is the free energy, and what are the features of the free energy that I can identify. In other words, you can see here the state 3, which is fully polarized, low in free energy, which means that it's probably the fullest state. And the state 1, which has another minima or dip here, is slightly the unforced. I'm just showing it for a 3 residue peptide because it's easier to demonstrate, but you could do this for any number of residues in the protein. In other words, even if you have a 500 residue protein, you should be able to generate all of these particular descriptive points. Right. So with this in hand, the model has certain. I'll just move it to the next slide, which is the model. What it takes in as an input is that it takes the structure of the protein. The RCSB or the protein data bank has the XYZ coordinates of every residue or atom in the protein. Now with that in hand, now I can construct something called the van der Waals potential. I can also construct something similar to a Debye-Heckel-like potential. I can also calculate some kind of way to estimate what is the correlation parameters here and the entropic penalty. And hence I can have some kind of stabilization free energy for each of the microstates. For every microstate, I will have some free energy value that I can derive from the protein structure alone, without worrying about all the different states that are potentially possible which will generate in molecular dynamics. Right? So what we did was, now we have this particular protein Cnu that I talked about. And what we did was we took this particular protein, we generated all these different microstates, we calculated the stable or rather the free energy of every microstate, and then we tried to reproduce some of the experimental observations. But to reproduce experimental observations, you need to have an experimental probe that gives you some unbiased information about it. And the primary probe that we use that can give us information on the unfolding transition is something called the heat capacity. And from StatMech, you know that heat capacity profiles or heat capacities are effectively measures of thermodynamic fluctuations, enthalpic fluctuations in the system at constant pressure. And this is a function of temperature. We have experimentally measured the heat capacity of this particular protein as a function of temperature. And what you see here is that at low temperatures, the heat capacity is lower, and at higher temperature, the heat capacity is higher. What it suggests is that the fluctuations are lower here, the fluctuations are higher here. And if you look at a protein that is at low temperature, the protein is usually folded and the fluctuations are very minimal. But as you go to higher and higher temperature, the protein starts unfolding and you can sample many different states or microstates. And that's why the heat capacity is higher here. And this is an actual experimental data. And what you can see here is that at the lowest range of temperatures, which is about 280 to 310 Kelvin, you have a very steep rise in heat capacity, which means that protein is not rigid. It actually starts sampling many different states in the nuclear ensemble. And this is some reference line which is called the Rayleigh baseline. I don't want to get into this, but this is some empirical relation that tells you what is the extent of fluctuation that you need in, observe in your system if the purely based on the molecular size. In other words, as a function of temperature, this is the expectation that the protein behaves like a solid structure, doesn't fluctuate at all, very rigid. And this is the way you would expect this protein to behave as a function of temperature. And it's not unfolding. And here, what you see is that you see an increase in enthalpic fluctuations as a function of temperature, suggesting that the protein is not rigid, it's sampling many states. What are these different states is what we do not know. And one other way to compare it is to look at another system which has been established to be a very rigid system. The sense that does not change its conformational fluctuation significantly. The blue is the CNU protein that we are currently studying or talking about, and ubiquitous 76 residue, which you'll be aware of, and it shows very little fluctuations in the range of temperature. And you can see that it has very little fluctuations out here. It's very flat and matching the Rayleigh baseline, and it has the same TM as this particular protein has a very different behavior. You can see the steep rise in pre-transition. And it suggests that CNU is a protein that you cannot call as a two-state system, meaning it's not sampling folded and unfolded states, it's probably sampling many different states in its radio ensemble. And that could potentially drive its functional response. It's our kind of hypothesis.
But if you go down the temperature much lower than 280,
right?
You will find it behaving like exactly like that.
Exactly. Yes. And you can actually see here, as you go to lower and lower temperatures, it kind of matches with what you would expect for a rigid system. In other words, the temperature itself is a thermodynamic variable that this protein is sensing. So, and that results in an enhanced fluctuation. The protein has been designed to sense temperature, and hence the range here is able to undergo enhanced fluctuation.
And if you go very far away, beyond 400 degrees,
yes,
how the behavior will be similar?
That I do not know. We can go all the way till about 400 Kelvin.
Transition going maximum and then going down.
It should effectively, and beyond a certain point, it should behave like a straight line, if I can say, the protein molecule. But we haven't gotten that because there is an experimental limit to that. So you will not be able to get that. So, for example, this Rayleigh baseline that I'm talking about, which is a measure of fluctuations in the rigid system, is calculated only in this particular range of temperatures because that's where the protein is stable, and that's what people are.
Is there any understanding on the transition, thermodynamic transition?
Yes. So
the order of the transition, like first order, second order.
Right. So the first order, second order, we believe it's a combination of first and second order. That's what I'm going to come to in the next slide. And
For that, you need some specific heat or something.
Yes, so this is heat capacity. We already have it. So the molar heat capacity we have it. We can also deal in terms of specific heat as well. So this information you have here. Now what we do is that since we have the total partition function of the system, for example, if I go to the previous slide. So by looking, if I take this particular protein CNU and I can calculate all these different states, I can calculate the statistical weight of each of these states, and I can calculate the partition function of the system. And once I calculate the partition function, what I can do is I can kind of calculate heat capacity as a derivative of the partition function. And once I calculate this heat capacity, I kind of superimpose on top of this. Because to calculate the partition function, I need all the different energies that I'm considering. I need to know what are the van der Waals interaction energy that I'm considering, what is the solvation term, what is the electrostatic term. All of those I can get it from the structure and also calibrate it with respect to the model and hence kind of fit the data. This is not a prediction from the model, but I'm using the data to calibrate those parameters and then I get this particular fit from it. Now, from this, since I have calibrated the model, now I can go ahead and predict what the free energy profiles are, right? And that's what I do in the next slide. So after doing this, so now we have some understanding of how this particular system behaves based on this, and we are able to explain the experimental data. And this is what we see when we plot the number of structured residues, which is the order parameter, which tells you the progress of the reaction as a function of free energy. So the x-axis, the more to the right you are, more folded is the protein. Larger is the free energy, more unfolded or more unfavorable is that particular macrostate. Here you can see that the blue is at 278 Kelvin, which is about 5 degrees Celsius, and you can see that unfolded like conformations, which is low values, are very high free energy. You can see the shooting all the way up there. And but the protein is fully folded out here. Right? Meaning this is a guy that is fully nicely populated. But now, so increased temperature going from green to red and then to black, you can see that there is a small movement in the native ensemble, the folded state. And you can see that the folded state is actually not fixed here, it moves from here to the left. And this can be observed by converting this free energy to probability densities. And what you see here is that the lowest temperature, this is the folded state that you observe, but there is also this partially structured state that you see, which is, we call this the N star, which is the partially structured state. And this is like a second order transition, because we don't see a dramatic change in the free energy. But between the partially structured state N star and the unfolded state here, we have a barrier, and hence we think that's the first order. So it's a combination of probably a first and second order unfolding that we see for this particular system, right? So, and this is quite interesting because when you imagine a protein that kind of is able to sense temperatures, you want this to be in a switching on and switching off with respect to temperature conditions. So when the temperature goes down, you want the protein to be fully rigid. And when the temperature slightly increases, you need to have some kind of higher flexibility, higher fluctuation, and hence it can perform differently. When the temperature rises even more, it needs to be more unfolded light, and hence it will need to function even differently. So which means that you need to have some kind of graded response to the temperature. It should not be just 1 and 0, but it should have a graded response to the temperature. Most systems, most bacterial systems, most proteins behave in the manner. They all respond in a gradient like manner. And we think that's what is happening here. So the N is the folded state, N star is the unfolded light conformation. We'll come to what it means. And this fluctuations here might likely drive the sensory response of bacteria. And I think that's what's happening out here. And this is something that I don't want to get into for this particular talk. But now what we can also do is to take a residue index, meaning we map it, the probability of every residue to be quoted as a function of residue index. And you can see that at the lowest temperature, this fourth helix has a lower probability. In other words, this is the fourth helix that has a lower probability. And this is the probability at different temperatures. This is the lowest temperature, this is the highest temperature. And you can see that this region, the first 3 helices are fully cooled. That corresponds to this helix 1, the helix 2, and the helix 3. All these 3 helices are fully cooled. But the fourth helix is unfolded even at the lowest range of temperature. That's what we observe here. And we also did analytical ultrasimplification. Sure. What happens when you change temperature? Effectively, is a technique that tells you what are the dimensions of the system. So basically, if your protein is rigid, it will sediment faster. If it is more expanded, it will sediment slow. It's simple logic, right? So based on this, we can argue that at the 278 Kelvin and 298 Kelvin, it's an experiment, and you can see this is a measure of the slope gradient of the protein at 278 and You can see at 270 it has a smaller stroke radius, in other words, it's more spherical, let's say. But as you put a higher temperature in the spherical approximation, still you have a higher radius, which means the protein is sampling more states, kind of swelled up and sampling more configurations. And that corresponds to the higher strokes radius. And we think this is happening within the physiological range. These are experimental measurements. Right?
But the sedimentation can also depend on the shape of the protein.
Yes, yes.
So because something is oblong,
Yes, or spheroidal, or yes.
something is
Prolate.
it will also take more time to set.
Yes. So, and these are evidences, right? So the idea here, the way we do the work is that we try to give as many evidences as possible, that this is what it is. Of course, we have to assume some kind of approximation as to what shape we are looking at. Of course, we do not know the shape. We just have this as the particular experimental issue. The shape can vary, but assuming a spherical approximation, this is what it means. But coming from DSE, coming from the tryptophan probe, because we saw from tryptophan probe that this region was already melting earlier. So based on these, we think that this particular region of the protein is actually melting earlier, and that results in the transition from N to N star. N meaning this entire protein is fully folded, the native structure, but N star is a state where the 3 helices are fully folded or nearly fully folded, but this particular helix is partially structured in solution. And when this is partially structured, you can sample more states out here, and hence the stroke radius will rise or increase. That's what our hypothesis is, right? And we also did NMR, and this is in collaboration with David Fushman at the University of Maryland. For some reason, the solution is not great. And this is a HSQC spectra. I don't want to worry about this, but the important thing that I want to say here is that this is the without full assignment of this particular spectra, we can look only at the tryptophan indole region. The indole ring of the tryptophan has a specific resonance that you can monitor, and because there is only a single tryptophan at this particular protein, we not assign this particular protein also because there are many conformational states, meaning the peaks are not well resolved, and that's because of the conformational exchange between many different states. And what you see here is a 283, 296, and 310. I guess it's 10 degrees. Sorry about this. I always prefer Kelvin. I never convert it into degrees Celsius, so we just keep it that way, right? So what happens is that you can see at 283 Kelvin, the tryptophan, this is the tryptophan indole region. Tryptophan is already sampling many states. Ideally, for a protein that is fully folded, the tryptophan should be rigid, should sample only one microenvironment, and that's going to give rise to one specific resonance out here. But what we see here is that the tryptophan, even at the lowest temperature, is giving rise to many resonances. At 296, the number of resonances decrease actually. And as you go to 310 Kelvin, what happens is that it still decreases. That's likely because the exchanges become faster at higher temperatures because the protein is sampling more states, and hence these kind of average, and there's conformational broadening, exchange broadening that contributes to this particular disappearance of resonances. And we think this is what is happening. This is a direct evidence, like SEC or WSME model based analysis, which are indirect evidences that we get. So this is a direct evidence, and hence we are fairly confident that this is what happens. And we think the whole overall storyline is that I don't want to get into this. We also did MD simulation, but the idea here is that at 280 Kelvin, this particular region of this protein is fairly rigid, and this is the N state which we labeled as the N native state in the model. But now, as you go to higher temperatures, what happens? This is the position of the tryptophan as a function of all the different frames, and you can see that this tryptophan is sampling many different things. Just like a creature that kind of evolves. So when you go from 280 to 310 Kelvin, and this region is now able to sample many states, and hence corresponds to the increased dimensions of this particular protein, explains the quantum yield data, and also the sedimentation velocity data, and also the WSB model prediction is validated. Some principal component analysis, worry about this, but this is the overall idea. So we think that this is what is happening, and hence, depending upon the fluctuations or excursions of this particular helix, conformational excursions of this helix, the protein is able to kind of sense temperature conditions. But of course, it wouldn't make sense if it just senses temperature. This sensing of temperature by these conformational fluctuations need to be translated to something else. In other words, when you take this structure and you go to the literature and study where is this particular protein binding to H-NS, this other protein that I talked about, which is the repressor, this is exactly the binding region that it binds to the repressor. So what it means is that at lowest temperature, this region is fairly rigid, and hence it's able to make a nice interaction or enable a nice interaction with this particular H-NS molecule. But as you go to higher temperature, what happens is that since it samples many, many different states, it's not able to form a stable complex repressionist, and hence this repression is not possible. And that's what we think is happening as well. And this is just a summary of this work, and I don't want to get into this. But what we talked about until now, or what I talked about until now, is only on the temperature sensing. But we believe this is also acting as an osmo-sensing in the sense that it also probably responds to changes in the solvent conditions when it comes to osmolarity, or the strength of salt concentration that you have in solution. Right? So, and this is the electrostatic potential map of this particular protein. You can see that some regions are highly frustrated, in the sense that they have a large number of negative charges that are very close to each other in space. This is from a solid structure. And in the other part of this particular protein, there are a large number of positive charges that are close to space. Generally, when you have systems like this, many proteins have this particular behavior. Generally, when you have this particular behavior, you have strong sensitivity to salt. Because what happens is when you have this particular protein folded, when you add more and more salt, you're going to shield all these interactions, and hence the protein might get more stable, or it might get less stable, depending upon how much of these interactions are coming from local interactions, how much of these electrostatic interactions are coming from local or coming from non-local sequence centers. Right? So, again, so we just keep it simple. We did this particular experiment, heat capacity measurement at different salt concentrations, all the way from very low salt to some kind of salt concentration or adnexal condition which mimics the insides of a bacterial cell to very, very high salt concentrations of 500 millimolar. We don't see dramatic changes in the heat capacity profile, except for the fact that the stability, which is effectively a measure of this particular peak position, increases or shifts to the right.
So the charges are separate, positive and negative charges are separate.
Separated for some reason in this particular system.
So this whole thing will act like a dipole, electric dipole.
Yes, yeah. And this is something you see here in this case. I wouldn't want to get into this, but all I want to focus, take a focus to is this particular slide, where as a function of wavelength, I'm showing you the near UV CD signal, right? Near UV circular dichroism. This is a measure of the tertiary structure of the protein, right? What happens is that when you go to low salt concentration, which is 43 or 90 millimolar, this is the spectrum at 5 degrees Celsius. But now as we increase the salt concentration to 170 millimolar or 500 millimolar, the spectrum is completely shifted. So it suggests that it's very difficult to understand how the near UVCD signal or where it comes from, because it comes from the coupling between the transition dipoles of the tryptophan with the backbone, protein backbone. There's no ad hoc prediction software that we can use to understand what's going on, or basic understanding from the perspective, for example, chemistry of what's going on there. But based on this observation, we can suggest or indicate that when you change salt concentration, the structure of the protein undergoes non-intuitive changes. And we believe that this is probably also the reason why this particular protein potentially can sense changes in salt concentration. And we can also do the same experiment, which is NMR, as a function of different salt concentrations. This is at 13 degrees Celsius, this is HFQ spectrum, and this is at 37 degrees Celsius. And you can see at 14.5, there is large conformational or exchange broadening of it, which means that generally when you take a spectrum of a protein at the lowest temperatures, what you will see is you will get sharp resonances for every residue, assuming that the protein is going to there. But within the NMR time scale, there are many states that are constantly exchanging with each other. What will happen is that you will have conformational broadening, and you will see the spectra being not very sharp. And you can see this at 14.5 millimolar, meaning at very low salt concentrations. Likely, what happens is that since I showed you that there are several positive charges close to each other, several negative charges close to each other, at low salt concentration, there is less shielding, and the protein is unable to interact or rather fold fully, and hence it's sampling many states. That's our hypothesis here, and you can see this in the NMR spectrum. But as you increase the salt concentration, the resonances become more sharper, which means that it's kind of all these different conformations are kind of coming together and giving rise to one specific conformation. But this can be counteracted by temperature. At temperature, both the proteins at 37 degrees Celsius, both the proteins behave. Both the salt concentrations at high temperature, protein behaves the same. Right? And again, the idea here is that at low salt concentrations, this is what we think is happening. These different cartoons that are kind of superimposed here coming from simulation, and the protein is actually samples many different stages for a rigid structure. And as we increase the salt concentration, it kind of becomes rigid because this And this is what we think is happening. And though, of course, the port helix is already sampling many different states, irrespective of what salt concentrations you are at. Right, and we did some implicit solvent empty simulations that kind of gave rise to or gives rise to a similar behavior. At low salt concentrations, you can see two different substrates that are populated. As you go to higher salt concentrations, you have the port helix that is still unfolded or partially structured, but it's not as heterogeneous, this particular distribution, as compared to this particular state. Right? And as I just told you before, it's not just CNU that acts alone in solution or inside the bacterial cell, but it also interacts with H-NS. And this particular interface, which is the port helix, is the region that binds with H-NS. And we had lots of trouble in purifying this particular protein, H-NS. It's a very sensitive protein, and we were finding it extremely difficult to purify this protein. We somehow managed to purify only the N-terminal domain of this protein. I will come up with the structure in a later set of slides. But when we purified this protein and we titrated it with CNU, what you can see here is that this is the fluorescence emission spectrum of CNU as a function of wavelength and in the free or in the bound form. In the sense that when you have the protein that is free in solution, the absence of H-NS, you have a red-shifted emission profile. But as you add H-NS, what happens is that is the bound CNU. You can see evolution, which means the tryptophan is a more varied environment because it's the region where the protein binds to H-NS, and hence you see evolution and also an increase in the fluorescence intensity. So with this, we were able to actually monitor the binding reaction. And when we titrated this, what happens with H-NS, which we have in solution, this is CNU that we have in solution, which has a tryptophan, and we titrate increasing concentrations of H-NS, this N-terminal domain alone of this H-NS of this, and at different salt concentrations and at two different temperatures. And you can see at low temperatures, you can see very distinct binding equilibrium, but at higher temperatures, the binding equilibrium is slightly different. But the important thing here is not just the difference in the binding equilibrium, the inflection points will give you some idea of the dissociation constant between the two molecules. But beyond that, what happens is that if you look at the low salt concentration, you can see it's a sigmoidal-like profile here, and the same goes for red out here. But when you look at the blue here, it's significantly sharper. And this sharpness suggests that there is not just one-to-one interaction, but there is a higher order term that is involved, likely from two molecules of H-NS interacting with one molecule of CN. And we did some analysis again. I don't want to get into the details here. The important thing is, as a function of salt, the kind of binding equilibrium between CN and HS varies dramatically. What the idea here is that, and you can see this is NH again. I think it's a projection which is having an issue here. This is the Hill cooperativity coefficient. Generally, for 1:1 complex, you will have this value around 1. But as you go to low salt concentration, what you can see is this value is about 1.5, which means that when you have a non-integer, what it suggests that there are many different kinds of complexes that are present. It's not 1:1, it can be 2:1, it can be 3:1, we do not know what it is. It can be any combination. But we think that's what is happening here, and that varies. The extent of complex formation also varies as a function of temperature. Right? So, and again, I'll skip this slide because I have more detailed slides in the next one. Right? So now, CNU, we have shown already, it's a sensor of temperature somehow, and based on this C-terminal helix or the fourth helix that unfolds at different temperatures. But we also have CNU sensing or responding to changes in ion exchange conditions, the kind of salt that we put in. Even we had some experiments before which I didn't show that depending upon what kind of salt that we add in solution, the protein must be behaving different. So that is something we observe. But of course, in most systems, it's not just one protein that is going to be responsive. It's a combination of proteins that interacts and kind of response to many different features here, whether it's pathogenic response, silencing of transcription, or compaction of DNA. Generally, what happens is that when bacteria are stressed, they kind of compact their DNA such that they're not expressing. They go into some kind of phase that they don't express any genes. Right? So that's what happens, and we think that's what is happening here as well. But it's not just a simple reaction as the way I had kind of explained in the previous set of slides. Imagine if this is H-NS, which is fully coated, and I'm showing you the circle here. As a function of temperature, pH, or osmolarity, it might be that the H-NS itself undergoes changes in the polar equilibrium. There may be changes in low temperature to high temperature, the H-NS itself unfolds, and hence it's unable to repress genes. Or the There's something we discussed before. It can undergo oligomerization reaction, which is very sensitive to changes in temperature or pH. Or it can, the binding to DNA can be sensitive to the DNA shown here in this particular purple color. That can also be sensitive to pH, temperature, and so on. Or it can be a combination of all of these three, which you have here: monomer to oligomer to an unfolded monomer to the binding to DNA. Again, all of these reactions can be controlled with changes in pH or temperature. But inside bacterial systems, we don't have just H-NS. There is also another protein that competes with H-NS to interact non-specifically with DNA. These are called nucleotide-associated proteins. H-NS is also a nucleotide-associated protein. There are other proteins though that can also compete with H-NS. These can also be determining what happens to the stress response additions. Of course, finally, you also have these co-repressors that can also regulate this particular region. So it's not just one protein molecule interacting with another protein molecule. It can be any of these. And I think this is what is happening in reality within a bacterial system. I'll show you why this is quite interesting or more important. Right? I told you that we weren't able to purify this particular protein H-NS. This H-NS is shown in the circles here. And the CNU that I talked about is this particular protein, which is a co-repressor. Right? So we study the oligomerization of this particular protein. The simplest experiment one can do is something called the size exclusion. I'm sure most of you would know what you do is you pass the protein through a column that has multiple beads. The beads have their own particles as well. And what happens is the protein is a large molecule. What will happen is that it will not get trapped within the beads and will kind of escape out much earlier. But if the protein is small enough, what will happen is it will get trapped in the bead and it will come out at a later time. Right? So this is what we see here when you change the concentration of H-NS. This is the elution profile of this particular molecule. H-NS has a function of different protein concentrations. When you have very low concentration, the elution volume is on the higher side. What it means is that the protein is getting trapped inside the beads of this particular column and it's eluting only later. But now as I add more and more protein, in the sense that it's a very high concentrated protein, let's say 200 micromolar, what happens is that not only is the shape kind of different, it's actually shifted extremely to the left. In other words, protein has grown into a big monster and hence it's not able to go into the beads and it's actually eluting out earlier and coming out at about 160 ml. Now, this suggests that H-NS undergoes concentration-dependent oligomerization at the range of temperatures. Here, we used about 300 millimolar additional conditions because that's what we can do for a column of this size. Right? So, and of course, this one observation can be some other parameter which is wrong, some error in the experimental measurement. But because we know the structure of this particular protein, what we were able to do is identify the regions which oligomerize, and we mutate this particular regions via specific mutations by eliminating salt bridges that are present in the system. And we have D68N and D71 mutation, and we do the same range of concentration for this particular mutant, and you can see, though you do see some concentration dependent on the solution volume, you see it's not as dramatic as what we see here for the wild type, suggesting that H-NS indeed something been known for a long time in the literature, but no one knew what the molecularity is. We'll complete in the next slide, and we think that's what is happening. In particular case, we are able to identify these mutations, and we also have a phosphomimetic mutation, natural phospho group that is introduced into this particular protein to eliminate this oligomerization, and we find even sharper profiles of it. This is a very little shift in the elution volume, right? So this tells you that it undergoes concentration-dependent elution or rather concentration-dependent oligomerization, but it doesn't tell you whether it's a monomer, dimer, or again, we resorted to the analytical ultracentrifugation that gives you an idea of the different species that are present in solution. And when we did this at say 15, 50, or 125 micromolar, what we get is very complex sedimentation coefficient or distribution. In other words, this will tell you some measure of the dimensions of the particles that are present, and this tells you some measure of the probability density, right? So what you see here at 15 micromolar, meaning low concentrations of the protein, as per the size exclusion chromatography, you know that there are smaller molecules that are present, smaller structures that are present, and you can see that green stops here. In other words, you have only lower sizes of the species that are present in solution. But as you go to higher concentrations, you can see that the infrastructure or species that is coming here. We think that is coming from the fact that this is a monomer likely, this is a dimer, This is likely a combination of tetramer, maybe a trimer, we cannot say for sure. And this is probably an octamer that is populating. So you have not just a monomer, but you have dimer, tetramer, and octamer that is present in solution. So it's not just the folded is an unfolded equilibrium that you need to be worried about in H-NS, but also the temperature dependence and salt dependence of these different species that you have present, apart from the way you drive the equilibrium between folded and unfolded that you need to worry about. This is at 150 millimolar Einstein condition, which is something like a physiological Einstein, let's say. But when you do it at 300 millimolar, you can see similar behavior. But say at 200 micromolar of protein, what you see is that you have a monomer and you have only an octamer that is present in solution. It's pretty interesting because at lower concentrations, we have monomer, dimer, and a tetramer. But at high protein concentrations, you have monomer, very little of anything else in between, and a lot of this tetramer that is present in solution. The protein is actually very, very sensitive to salt concentrations and kind of salt. Unfortunately, we couldn't do much more than this because we are collaborating with the group in Japan on this, and this has come up after a couple of years of constant pestering them.
Do you have any understanding of why it is happening?
Why it is happening, and that is coming from salt dependence, we do not know. There's seems to be some degree of cooperativity here that we see here. Monomers or an octamer.
Smaller number or large number?
Yes.
Nothing in between.
Yes, that suggests certain degree of cooperativity associated with it. The moment you have 2 or 3, it won't stick. It won't stick to 2 or 3. So that's what seems to be happening at higher protein concentration, but lower protein concentration, it still seems to form monomer, dimer, and a tetramer.
Is it possible to go beyond that 200?
We can do that, but we were restricting ourselves to about 200 simply because that's about the physiological range, maximum concentration of H-NS that you find within bacterial cells. So that's about 200, 150 to 200 micromolar the concentration. So we kind of restricted ourselves to that. And we had problems in this collaboration where we send the sample out and then it takes us about 8 months to get the data from them. Yeah, after several reminders and stuff, they were very happy and initially very nice to start with to initiate the collaboration, but we never went anywhere. So whatever we have, we are very happy to achieve. So we stopped there. It's a group in Japan. Uchiyama, very nice person and world leader in centrifugation and analytical alpha centrifugation. But unfortunately, collaborations you need a lot of personal sync, and that was difficult. But we are glad. We are currently waiting to get information on the mutants, and we believe we will see only this and this peak for the mutants, and not any of the other peaks that corresponds to higher order oligomeric. Right?
So monomer and dimer alone.
We think that's what is going to happen in the mutants because we eliminated higher order oligomerization, but the dimerization should be present because that site we don't touch as per the mutation. And it is just a cartoon representation of what we think is happening as we go from low concentration to high concentration. Every circle corresponds to a H-NS molecule, and we think at low concentrations it's likely this is what is happening. If you go to higher concentration, this is what is happening. If you go to even higher concentration, this is what is happening. H-NS is very interesting again for something which I missed out is the fact that it controls about 75% of the stress response genes in bacteria. So any stress response, H-NS is the first line of defense. So it goes and kind of represents all the genes, keeps everybody happy. So we think this completely because of the fact that it's able to oligomerize in a very interesting manner. I'll just have 3 more slides. I'll stop there. I believe I have 3 more slides. I don't remember. But now we also wanted to study what happens with the binding to DNA. Okay, this is not holding, it's not just binding to protein to itself, homo oligomerization, but it's also hetero-oligomerization because H-NS is supposed to bind DNA and repress the expression of genes. So we did a simple anisotropy based experiment as the function of H-NS. In other words, what we do is we take a DNA, tag it with a dye, and monitor the tumbling time of this particular DNA as the function of this higher protein concentration. When the protein binds more, the DNA binds more and more protein, the tumbling time will slow down, and hence that will result in an increase in anisotropy. Right? So what we see here is that just focus on 25 degrees, that is sufficient, and wild type is the color mismatch here. Fine here, but here it seems to be different. This is the data for wild type at 25 degrees, this gray circles. As you add more and more H-NS, more and more H-NS binds DNA, and hence the tumbling time slows down, and anisotropy starts decreasing. It's not just a sigmoidal-like function. You can see a couple of phases out here in a certain sense. But beyond a certain point, what happens is that this anisotropy starts dropping. So, and what we think is happening here, the dropping of this particular anisotropy simply because of the fact that now instead of binding DNA, it starts oligomerizing with itself. So here you have hetero-oligomerization between protein and DNA. But beyond a certain point, homo-oligomerization with H-NS is effectively eliminating binding to DNA. I think that's what's happening out here. And one simple way to test this is by using the double mutant. The double mutant is not supposed to have higher order oligomeric structures, right? Because we have eliminated higher order oligomerization, it will form only tetramer. And when we add this, what happens is that we see a similar trend, but the dip that you see here is not as strong as you see in this particular case for the viral. So we think that's what is happening in terms of the competition not between just folding, unfolding, oligomerization with itself, but also the degree of oligomerization with itself and with DNA. So there is one more layer to it. And phosphamimetic mutations of phosphorylation can also control the extent to which it oligomerizes. And I believe that's also happening. So again, we did this as a function of temperature. I don't want to get into it. Again, it's just cartoon representations of what is going on. So at low, I don't even remember what it is. Yeah, so at low concentrations of the protein, what happens is the DNA is free. As if you add more and more protein molecules in the solution H-NS, it binds DNA. But beyond a certain point, what happens is it prefers forming these octamers rather than binding to DNA. But when you take the mutant is able to nicely bind DNA at different concentrations, respectively. What the concentration is, because it does not undergo so much of oligomerization, right? And we also measure the heat capacity profiles of this particular thing. Last 3 slides, I promise. So the heat capacity profiles at these different concentrations of the protein, and what we see is a very interesting behaviors. Again, I will not go into the details. The important thing is that when you change the concentrations, these features are extremely interesting because you can deconvolute these heat capacity profiles. We love heat capacity profiles, right? So, and when we can deconvolute this using statistical thermodynamic arguments and get the populations of the different states that are probable in solution, right? So there are some observations that you can make that I kind of skip. But the important thing is that now I can write a simple thermodynamic equation out here. It says that I can have these different kinds of equilibrium that is possible in solution, right? The monomer can be in equilibrium between an unfolded state and the native state. Native state means fully folded state. And the native state can be a dimer. And hence I can write a dissociation constant for this, or it's an equilibrium constant between an dimer versus 2 of this. Basically 2 monomers coming together to form a dimer, and hence you will get this square term up here. And I can also define an oligomerization equilibrium constant, which will consider the higher order oligomerization, right? And since we see only about octamers, we just use this in particular equilibrium. And now with the mass balance equation, I can solve for, since I know the concentration of C, I assume values of K0, KD, and KU that is sufficient enough to explain the experimental data. And I get the values of K0, KD, and KU. And I also build a partition function for this particular system. Okay. These are details. And I can calculate the fraction of the different species that are present based on the partial partition functions of this particular system. In other words, if it's an octamer, I know what fraction of the molecules that are present in solution is an octamer, and if it's a tetramer, what fraction of species that are present in solution is a tetramer. By quantitatively or semi-quantitatively reproducing the heat capacity profiles, because I can reproduce heat capacity profiles using the partition function by taking the derivative of this heat capacity partition function. Right, and when we do that, again, we did this in a very semi-quantitative manner, and we were able to predict the population of the different species that are present in solution as a function of temperature. And what you see here, you can focus just on 15 micromolar. What happens is that the octamer is in low concentration at 15 micromolar. This we remember if you go to the AUC slides, analytical ultracentrifugation, the highest peak was not visible. Only at a higher concentration, you had the peak that corresponds to the octamer. Right, so you can see the octamer population is extremely low, and this population goes down with temperature. The tetramer and dimer are more populated at low concentrations. And now what happens is that as you increase temperature, the tetramer goes down, the dimer goes up because of the fact that octamer and tetramer are melting. The dimer population goes up. But now, as you increase the temperature more and more, what happens is the protein starts unfolding. Right, so what happens is the dimer population goes down, the monomer goes up, and hence the unfolded state also goes up. That's what we think is happening. And this coming to this cooperativity issue, and you see some evidence of this here in the 200 micromolar. You can see there's very little population of tetramer and the dimer out here. There's a lot of this octamer that is present. and the octamer is about 60% at this particular condition. And now as you increase the lowest temperature, now as you increase the temperature, the octamer drops, tetramer increases, dimer increases, and eventually beyond a certain temperature is the monomer that increases, and eventually the monomer also starts unfilling, and hence you see this way. So we think it's a very complex system which cannot be simply classified as monomer, dimer, tetramer, octamer. It also depends on how it competes with DNA, how it unfolds, and how it interacts with this co-repressor. So like multiple layers to it, and which can be regulated by not just these conditions, but also using post-translational modification, particularly phosphorylation. I think that's what is happening, though experiments are still underway to understand what is going on. Okay, thankfully for you guys, I guess. So, and the idea here is that H-NS exhibit a complex equilibrium, which is extremely complicated, hence probably explains why we were unable to purify it in a reasonable sense. Right, and the oligomerization status, we have some preliminary data, but we don't have confidence in this. I'm showing you here that can also be modified by its co-repressor. Cnu itself exhibits temperature and osmolarity dependent changes, and we believe that this is something very important. The equilibrium thermodynamic models are necessary generally in the field. This particular field, people show some biochemical evidence here and there and say, okay, and maybe arguments what's going on. We think that you need to have really an idea about what the stoichiometry is, what the molecularity of the reactions are, what are the KD values, the dissociation constant value, the changes in enthalpy and entropy changes that are associated with the system. And we believe the model that we again used for DNA binding is not completely right. Though I haven't shown you that, it's simply because when you have a H-NS molecule that's an octamer, there will be 8 DNA binding domains. They can actually interact non-specifically with DNA, and you can imagine combinations of these DNA molecules. They will not bind at the same site. They can bind at different sites. So you need to have very, very complicated models that needs to be come out to explain this. Which there are some models available in the literature, but we haven't tested some of them. Again, we are yet to understand this question of given a certain base pair, number of base pairs of DNA, how many H-NS molecules are actually found? We don't have an answer to this question, and what molecular mechanisms promote repression and compaction. At the level of hydrodynamic, we need to take about 1,000 base, 1 million base pair DNA, no, 100,000 base pair DNA to do these kind of experiments, which is again very, very complicated. For the protein itself, we struggle a lot, but I hope for the next 10 years, and when I come here in maybe 10 years' time, I give a talk, maybe I can answer this question on what happens in binds DNA. We believe this is a very interesting system simply because the reason why you're delving into great detail into the system is because generally, when it comes to antibiotics and the microbial resistance, what happens? There are antibiotics available for cell wall synthesis, peptide synthesis, DNA replication, and so on and so forth. But this is a very interesting molecule against which one can come up with drugs, antibiotics, or molecules that kind of eliminate its repression because they are very, very important for bacteria to repress the expression of genes or activate the expression of genes. So, we're able to use this molecule to develop drugs that could be a viable avenue to address antimicrobial resistance, or at least come up with antibiotics in slightly different direction. Because eventually in 10 years, 15 years, 20 years time, we are not going to have any of these antibiotics that are currently available in the market. In the last 50 years or so, there's no antibiotic that has entered the new antibiotic that has entered. So this probably could give rise to a collection of a class of antibiotics that could be quite interesting. But again, probably I need another 25 years to answer this. So I'm not going to commit myself to it. So with that, I will stop with acknowledgements. The work on Cnu was done by Abhishek, currently a postdoc in University of Alberta, and we see she's a PhD student. She worked on the H-NS and the double mutant H-NS, and Dr. Krea for IIT and H-NS, David Cushman for NMR, Chiama for the analytical ultracentrifugation. PhD students, some of them were graduated, stay as of the industry, and these are my collaborators across India and Spain, Italy, and funding from. Thank you.